This is a video to promote and to raise awareness for the three VICH guidelines, GL50R, GL55, and GL59, on waiving from batch safety testing of target or laboratory animals for veterinary vaccines. This video also shows the outline of seed lot system of vaccine, which is a prerequisite for applying these guidelines, in line with the typical production scheme of Newcastle disease virus vaccine production. There are three VICH guidelines, GL50R, GL55, and GL59, for waiving from test of target laboratory animals for veterinary vaccines. GL50R and GL55 are guidelines on waiving target animal batch safety tests carried out on every batch of vaccines. On the other hand, GL59 is a guideline for waiving laboratory animal batch safety testing of vaccines. First, to explain what batch safety tests are, we will give an outline of how vaccines are produced and how tests are carried out on every batch of vaccine produced. As an example, we will look at how the Newcastle disease vaccine is made using live viruses and embryonated eggs. First, the embryonated egg is inoculated with the production seed virus. After it has been incubated for a set period of time to ensure adequate multiplication of the virus, the chorioallantoic fluid is extracted to make a virus stock solution. A diluent and stabilizer are added to the stock solution. Then it is distributed into vials, freeze-dried, and sealed to make the final product. As shown above, vaccines are usually made using raw materials and incubation technology, so numerous quality control tests have to be carried out on every batch of the final product. Shown here are the tests that are normally carried out on the Newcastle disease vaccine, which we chose as our example earlier. They include safety tests on the chicken, which is the target animal. This test is known as a batch safety test, or BST. BST is the general name given to safety tests among the quality control tests carried out on each batch. BSTs are divided into tests carried out on target animals and on laboratory animals. Vaccine production uses viruses, bacteria, and other live microorganisms, as well as cells, animal serum, and other raw materials taken from animals, so there is a need to verify their safety every time. It has been mandatory for BSTs of vaccines to be carried out throughout the world. Here, you will need to note that the target animal batch safety test has been deleted from European pharmacopoeia monographs since 2013, and so is no longer required in the EU. Numerous animal tests are carried out in developing and inspecting drugs for animals. However, global advances in animal welfare policies have led to a need to minimize animal experiments. As a result, VICH is aiming to minimize animal tests, and a statement was issued in support of the 3R principles, namely the principle of replacement, refinement, and reduction, at its 19th Steering Committee held in 2007 in the U.S. Recent advances in manufacturing and testing technologies have led to greater stability and quality, resulting in debate on the need for BSTs. Decreased concern for adverse reactions from vaccines have been due to improvements in the quality of raw materials, culture purification methods, the assay method, the introduction of the seed lot system, GMP, and safety testing under GLP, and so on. BSTs are divided into two categories depending on the type of animal used, with those using target animals known as the Target Animal Batch Safety Test, or TABST. The GL50R and GL55 are guidelines on waiving these tests. When laboratory animals are used, the test is known as the Laboratory Animal Batch Safety Test, or LABST, and the GL59 is the guideline on waiving this test. The objective of TABST/LABST guidelines differ from the other VICH guidelines. VICH guidelines are usually those for test requirements for registration. However, the TABST/LABST guidelines are for vaccines that have been registered. Those guidelines indicate how TABST/LABST can be waived, and they have been adopted in accordance with the 3R principles. We will focus on GL55 in explaining the details of the three types of TABST/LABST guidelines. The details for the GL50R and GL59 are basically the same as GL55. Here are the contents of GL55. Section 2, shown in red, is the part covering the details of the guideline, so we will take a closer look at the important points. 2.1 Scope. This guideline is limited to the criteria on data requirements for waiving target animal batch safety tests of live veterinary vaccines. 2.2 covers regional requirements, while 2.2.1 explains the regional differences in requirements of TABSTs. 2.2.2 explains the other relevant requirements, which are GMP, seedlot, and pharmacovigilance systems for manufacturing control and early detection of safety problems. 2.3 explains the data requirements for waiving target animal batch safety tests. This is the most important section for the implementation of the guideline. 2.3.1 is an introduction explaining the three prerequisites for authorizing the waiving of TABSTs for the various vaccines by the regulatory authority. The three prerequisites are that a sufficient number of consecutive production batches have been produced and comply with the tests on specification, thus demonstrate consistency on the manufacturing process. From here, the actual regulations have been laid down for information needed for individual vaccines in order to waive the TABST. First is 2.3.1.1, the characteristics of the product and its manufacturer. The manufacturer should demonstrate that the product is manufactured following the quality principles, example, seedlot system and GMP. This is an explanation of the seedlot system. The quality characteristics of the master seed for the target product must have been investigated properly. It must be decided that batches of a product are derived at a given passage level. Other than those, manufacturing must be carried out in accordance with the GMP system. When other batch tests in target animals, like potency tests, are conducted during the manufacturing process, it is recommended to gain additional safety data in the tests. 2.3.1.2 Information available on the current batch safety test. The number of batch safety data needed to ensure waiving of the TABST has been indicated in this section. As a general rule, test data are required for 10 consecutive batches. But if fewer than 10 batches are manufactured in 3 years, the minimum of data for 5 batches shall be required. As a general rule, passing TABSTs for 10 consecutive batches will demonstrate that safe and consistent production has been established. Achieving this will require the following 3 conditions. 1. The conduct of the TABST shall be in accordance with the regional requirements. 2. Variety of local and systemic reactions should be examined. 3. Summary and discussion should be provided. Moreover, as a general rule, data from the TABST of combined vaccines may be used to waive the TABST of vaccines containing fewer antigen and/or adjuvant components, provided the remaining components are identical in each case, and it is only the number of antigens and/or adjuvant which has decreased. For example, TABST data from a combination product can be sufficient to waive TABSTs for all the fallout products. The manufacturer should provide a summary and discussion of the findings. 2.3.1.3 Pharmacovigilance data. A pharmacovigilance system in accordance with the VICH guidelines is in place during the consecutive batches where on the market. The data to demonstrating the consistent safety performance of the vaccine in the field should be provided. Section 2.3.2 lays down the procedures for waiving the target animal batch safety test. A report should provide an overall assessment of the consistency of the product's safety and would include the number of batches manufactured, the number of years the product has been on the market, the number of doses sold, the frequency and seriousness of any adverse reactions, and investigations into the causes of these adverse reactions. Last is an explanation on the application of these guidelines in Japan. GL55 has been implemented in Japan from April 2018. Applicable vaccine. The vaccine which has fulfilled all the following requirements. 1. Live vaccine for veterinary use. 2. More than the latest 10 batches were passed by the TABST. 3. Seed lot system-based product. Application documents. Waiving the TABST requires applying for approval for procedural changes. The following materials are required when applying. Manufacturing records, the latest 10 batches. Batch release test data, latest 10 batches. Information on defect batch, if any. Revision history of the dossier. Rational explanation for waiving TABST. And overall safety assessment including pharmacovigilance data. The TABST may be waived if safety is verified upon screening. The VICH Guidelines 50R, 55, and 59 have already been applied to many animal vaccines, contributing greatly to animal welfare. This training material has been produced with the support of the Government of Japan.